Transport mechanisms across cell membrane. Table of content. Structure of cell membrane is composed mainly of proteins, lipids, and carbohydrates. A phospholipid is a lipid made of glycerol to fatty acids tail and a phosphate linked head group. Biological membrane usually involve two layers of phospholipid with their tails pointing inward, an arrangement called phospholipid bilayers. Membrane protein may extend partway into the plasma membrane across the membrane entirely or be loosely attached to its inside or outside face. And carbohydrate group are present only on the outer surface of the plasma membrane and are attached to proteins forming glycoproteins or lipids forming glycolipids. Types of transport: passive transport, active transport, vascular transport. Passive transport this takes place when molecules diffuse across the cell membrane by traveling through specific transport protein. It's occur down a concentration gradient. This means molecules move from an area of high concentration to lower concentration. This doesn't require a supply of energy because diffusion is a spontaneous process. An example of passive transport is the uptake of glucose by liver cells. Glucose diffuses from the blood into the cytoplasm of liver cells by a transport protein called GLUT4. Simple diffusion doesn't need protein carrier. Simple diffusion across lipid bilayer, very rapid for lipid soluble substance. Protein channels very slow for lipid insoluble, water soluble, charged, small molecular weight. Both lipid bilayer and protein channels very rapid for water soluble, small molecular weight, and charged. Facilitated diffusion doesn't need ATP, is a form of facilitated transport involving the passive movement of molecules along their concentration gradient, guided by the presence of another molecule, usually an integral membrane protein forming a pore or channel. Facilitated diffusion doesn't directly involving high energy molecules like ATP or GTP, since the molecules are moving along their concentration gradient. Cell membranes are only freely permeable to a very limited class of molecules. They must be small in size and non-polar, but facilitated diffusion can diffuse large molecules that doesn't diffuse through lipid bilayer like glucose, amino and nucleic acids, large ions, and large amount of water. Carrier proteins involved in facilitated diffusion often have two conformations. The bending of the molecules on the side of membrane induces a change in the three-dimensional structure of the protein, which allows the passage of the molecules through the other side. Factor affecting the rate of diffusion across cell membrane are membrane thickness. For nutrient to diffuse into a cell, they must traverse the cell membrane. Cell membranes are extremely thin to allow for the diffusion of materials across the cell membrane. Temperature and pressure. Increasing the temperature or pressure increase the kinetic energy of the particles, thus increasing the rate of diffusion. Concentration gradient. The greater the concentration gradient, the greater the rate of diffusion. Surface area. The greater the surface area over which diffusion can occur, the greater the rate of diffusion. Active transport. A process that involves the movement of molecules from a region of lower concentration to the region of higher concentration against a gradient or an obstacle with the use of external energy. During the process of active transport, a protein pump makes use of stored energy in the form of ATP to move molecules. The uptake of glucose in the intestine of the human body and also the uptake of minerals or ions into the root hair cells of the plants are some of the examples of active transport. There are two types of active transport, namely primary active transport and secondary active transport. Primary active transport. Most active transport is carried out by transmembrane ATPase, an enzyme that crosses the cell membrane. The sodium potassium ATPase enzyme is found in all animal cells. It maintains cell membrane potential by pumping 3 sodium ions out of the cell for every 2 potassium ion it moves into the cell. Secondary active transport. It takes place across a biological membrane where a transporter protein couples the membrane of the electrochemical ion down its electrochemical gradient to the upward movement of another molecule or an ion against a concentration of electrochemical gradient. How active transport works. Transmembrane proteins are proteins that across a cell membrane. They usually have one or more transmembrane demons that cross through the membrane lipid bilayer, as well as demons that bend with ligands inside and outside of the membrane. The protein recognizes the molecule or ion to be transported and passes it through its transmembrane demon. Vascular transport. It is the movement of macromolecules enclosed inside cell membrane bound vesicle. Endocytosis is a cellular mechanism where a cell internalises substance from the external environment. This substance undergoes certain process of breaking down to smaller elements either for use by the cell or for elimination purposes. Exocytosis is a process that is used to transport materials from inside the cell to the external part of the cell by use of energy. The mechanism uses special vesicles filled with the particles of interest to transport. Generally, in this mechanism of exocytosis, a special vesicle bound to the cell membrane containing the cellular particles will expel the cell content to the external part of the cell. Endocytosis transport. There are three types based on the mechanisms of particle include phagocytosis, pinocytosis, receptor-mediated endocytosis, calcineurin-mediated endocytosis. Mechanisms are used to elimination and such as amoeba use phagocytosis to trap and digest nutrient uptake and absorption of nutrient, a transferrin in receptor-mediated endocytosis to absorb cholesterol that is bound to bad cholesterol in receptor-mediated endocytosis. Example: white blood cells engulfing a virus and eliminating it. Exocytosis transport. There are three types depending on the sequence of process involved in transporting particles out of the cell. Include constructive exocytosis, regulated exocytosis, lysosome-mediated exocytosis. Mechanisms are used for transportation of proteins and lipids used to prepare the cell membrane after endocytosis. A transportation of glycogen and insulin hormones from the pancreas to the liver for breakdown and utilization by the body depending on body sugar level. Transportation of chemical signals between cells allowing cell communication. It is also important in synaptic transmission of information in the neurons. Example: releasing a neurotransmitter for cellular communication. Proton pump. It is an active transport process that transport hydrogen ion inside the parietal cell in the stomach that important for secretion of acid. Mechanism of acid secretion. The key player in acid secretion is a hydrogen ion potassium ion ATPase or proton pump located in the canalicular membrane. This ATPase is magnesium dependent. Hydrogen ions are generated within the parietal cells from dissociation of water. The hydroxyl ion formed in this process rapidly combine with the carbon dioxide to form bicarbonate, a reaction catalyzed by carbonic anhydrase. Then bicarbonate is transported out of the basolateral membrane in exchange for chloride. The outflow of bicarbonate into blood results in a slight elevation of blood pH as known as the alkaline tide. This process serves to maintain intracellular pH in the parietal cell. Chloride and potassium ions are transported into the lumen of the canaliculus by conductance channels, such as necessary for secretion of acid. Hydrogen ion is pumped out of the cell into the lumen in exchange for potassium through the action of the proton pump. Potassium is thus effectively recycled. Accumulation of osmotically active hydrogen ion in the canaliculus generates an osmotic gradient across the membrane that results in outward diffusion of water. The resulting gastric acid is 155 mHCl and 15 mKCl with a small amount of NaCl. Control of acid secretion. Parietal cells bear receptors for three stimulators of acid secretion, reflecting a triumvirate of neural, paracrine, and endocrine control: acetylcholine, histamine H2, gastrin.